These next presentations will combine Chapter 15 and 16 to cover the immune system. This first mini lecture will be on non-specific immunity, otherwise known as non-adaptive immunity. Before we begin, I want to remind you that there are quite a few animations available in the Mastering Microbiology study area for the chapters. It would be very wise to use them to help you with this complicated system. This is the body's first line of defense. In other words, the first defenses against infectious diseases. It's made up of things like structures, chemicals, and processes that are working to prevent pathogens from even entering the body. That includes our skin and mucous membranes, right? Both are a form of physical barriers. Also, our tear ducts, the lacrimal apparatus, and the normal microbiota, they play a role, among other things that you'll see on this next slide. In Chapter 15, Table 1 compares the skin and the mucous membranes. The skin provides a more formidable barrier for a lot of pathogens compared to the mucous membranes, and you can see some of the reasons why. Our other first line of defense, besides the skin and mucous membranes, includes production of antimicrobial peptides. These are short strings of amino acids that are secreted by our cells, and you'll find them present on skin and mucous membranes. The neutrophils also possess them and can secrete them. They have a variety of actions, but they all target microbial invaders and try to disrupt things like cell membranes so that they are definitely a defense mechanism. We also have enzymes like lysozyme, which are found in tears and breaks down cell walls. Then, the sebum in our skin that keeps it pliable and thus intact without breaks, and it lowers the pH, in turn inhibiting a lot of organisms. There are a lot of other chemicals and processes that are described in Chapter 15, Table 15.2. Now let's talk about the role of our microflora. The good microflora are those microbes that are helpful to us. They play a role in our first line of defense because they are antagonistic to microbes that are trying to come in and establish themselves on or in us. Because they compete with the pathogens for things like space and nutrients, it makes it harder for pathogens to get established. Therefore, the microflora physically prevents the pathogens from attaching or spreading. We also know that microbes compete by producing antimicrobial compounds. These compounds can work against the pathogens as well. Another important role of microflora is that some of our microflora provide vitamins to us, like vitamin K, that we do not produce ourselves, and that helps keep us healthy. Also, the microbiome stimulates our second line of defense, our adaptive immunity, which keeps our immune system on alert, and that's helpful in a lot of ways. Let's talk about the second line of defense. If pathogens or invaders succeed in getting through that first line, the second line of defense becomes activated. In this response, we're dealing with responses by cells and chemicals, and a lot of those things are found in our blood. We're about to look at the components that make up of our blood, and that, for the most part, helps prevent pathogens from becoming established. These components and activities are not something that our body has learned. They're already there, ready to work. When you think about blood, one of the things that happens within the blood components is that iron is limited by what's called sequestration. This means that the iron becomes locked up in proteins. These are 3 different proteins that sequester or lock up iron: ferritin, transferrin, and lactoferrin. Sequestration makes it very difficult for invading microorganisms to acquire iron from our bodies. And iron is an element, a trace element, that every living organism needs. Therefore, without it, invaders are unable to multiply. But pathogens often have ways to circumvent this and break apart these proteins to acquire the iron. Nonetheless, these iron-locking proteins exist for that reason, to prevent iron from being readily available to invading microorganisms. We also have this system called complement that we'll be talking about in a lot more detail later, but know now it is part of the blood. It is a protein activating system that is antimicrobial in nature. Then we have white blood cells, the leukocytes that are ready and able to respond. And some of them are part of our acquired immunity, which we'll be talking about later, but some are also just a part of our non-adaptive, our innate immunity. So when we look at our white blood cells, they're broken up on this figure and in many explanations, basically by how they look under the microscope. There are cells that are grainy looking under the microscope, and they're called granulocytes. Then there are cells that do not have granules, and they are agranulocytes. You'll see them listed down at the bottom, the different types of blood cells. We will be doing a virtual lab where you'll be looking at blood smears and identifying the cells by their appearance. Here, starting from the right, moving left, we have a lymphocyte. These are part of our adaptive immunity, so we will not go into details now. That'll come later. We have monocytes, which are immature macrophages that we find in our bloodstream. We have eosinophils, which are very red looking, as you can see. They're full of red granules, and they play a role in allergies and fighting off larger parasites. Next are neutrophils, which are a phagocytic type of cell that are considered the first responders. They arrive at the scene very quickly compared to the other cell types and attempt to contain the invasion. We have basophils, which also play a role in allergies, and as you can see, they're full of purple granules. Then I think you know what platelets do. They help with blood clots. And finally, here's our red blood cells, the erythrocytes. These are all the different cells that you find in the blood, and they originate from one type of stem cell in the bone marrow. Here's a micrograph of these different cells. We'll be visiting this topic in detail later. Basophils are the rarest type of blood cell. You do not find them very often, maybe less than 1 in 100 white blood cells when you are counting the types in a blood smear. Here are the eosinophils. Still not very common, but up to about 4% of the cells. Monocytes next. Going by abundance percentage-wise, you find them about 8% of the time. Neutrophils are very common, the most common, and up to 70% of the white blood cells in your blood are of this type under normal circumstances. And second to that are the lymphocytes, that can be around 1/4 of all of the white blood cells. When you're looking at the white blood cells under the microscope, you can anticipate how often you'll see these cells if you're looking at a normal blood smear. When you're sick, the total number of white cells in a volume go up, and the phagocytes increase as well. Allergic individuals can have high numbers of eosinophils, but high numbers of these cells can also occur if somebody is suffering from a parasitic infection. Taking a closer look at these blood cells, the granulocytes contain granules. They stain different colors based on the dye that you're using to stain. As mentioned earlier, the basophils stain blue or kind of purple. Your eosinophils stain red, and then the neutrophils stain more of a light lavender or lilac color. Interestingly, neutrophils and the eosinophils easily will move through the small openings or the small gaps between cells in your capillary beds. Other cells can do it too, but these cells tend to do it quite often. Neutrophils do it in order to phagocytose pathogens that are in our tissues. Look down at the capillary level down here. You can see that the cells are traveling through the smallest blood vessels, essentially getting one cell moving through at a time. This monocyte right here, if it detects something that's going on in the tissue because of chemical signals, it will become triggered and the monocyte will react by sticking to the side of the capillary. This is called margination, the sticking to the edge of the inside of the capillary vessel. Then the cell pushes its way through the small little gaps between cells, like you see right here. And that's called diapedesis. That's the cell that's migrating out of the blood vessel into the tissues in order to attack what's ever there. So remember these two terms: margination, the sticking, and the diapedesis, the migration. The second line of defense includes agranulocytes. Remember, the granulocytes look grainy under the microscope and the agranulocytes don't. Here we have lymphocytes and the monocytes. Lymphocytes are the ones that are really important for the adaptive immunity, and we'll be talking about them in the next chapter. Those include our B cells and our T cells. The monocytes are the immature macrophages, or rather, not activated macrophages. If there's a signal for the cells to leave the bloodstream and enter the tissues, monocytes will become activated and become macrophages. Macrophages are very good at phagocytosis and digesting up invaders. They're very important also in stimulating the adaptive immunity, although they themselves are not very specific. So they'll ingest pretty much anything, debris or foreign. Next, dendritic cells are important cells in our tissues. They really are a specific type of phagocyte with lots of extensions, a lot of arms touching nearby cells. That's why they're called dendritic cells, because they kind of look like they have dendrites, like a neuron, but they're a phagocytic cell with lots of extensions. They're a specialized tissue macrophage. There's lots of different phagocytic types of cells that are much like a macrophage, and they're named specifically depending on where they're sitting in your body. We'll end this section by looking at phagocytosis in a little more detail. You can look at this picture right here, seeing a phagocyte that is ingesting a yeast cell. We're going to break that process down into stages. Remember, cells that can do phagocytosis are phagocytes. This process is not completely understood, but a lot is known about it. Phagocytes in our own bodies are really important for stimulating the adaptive immunity, and we're going to come back to that, what's called the professional phagocytes, later to explain that process of activating adaptive immunity. Right now, let's understand the basic process. So the first thing that has to happen is a signal is triggered as a result from some tissue damage, or there might be some invaders nearby. Cells that are damaged will release chemical signals that then alert the phagocytes to move to that area. This response is called chemotaxis, which literally means moving towards a signal. The second thing that happens is that the phagocyte is going to attach to whatever it is that is the problem in that area. In this case, it was a bacterium that the phagocyte is going to attach to or adhere to and surrounds it with the extension of pseudopodia to form a phagosome. The bacterium is completely engulfed in that phagosome and is now in a little membrane-bound vesicle. Next is the killing. During this step, the lysosomes, which are filled with digestive enzymes, will fuse with the phagosome, forming a structure called the phagolysosome. At the time of fusion, those digestive enzymes are dumped into the phagosome. By doing this, the bacteria or yeast or whatever was ingested is going to be digested up and broken apart. Finally, the cell will take what it wants and anything that is useful, amino acids, nucleotides, whatever, and it keeps those things and it will throw out the waste. That is the elimination step, in which the phagocyte will exocytose anything that's not useful to it and allow the body to eliminate it as waste. We're also going to come back later and talk about this stage between here, the killing, and the elimination, where structures are made that can activate your adaptive immunity or your specific immune response. This is the explanation of phagocytosis, but it will tie in with adaptive immunity in a future section in chapter 16. This figure is showing you one of those interesting illustrations that your book has, where we have an actual transmission electron micrograph to show you the real thing happening on one part of the figure, and then the illustration on the other part so that you can visualize and more clearly understand how this process is occurring in real life. Well, here's where we'll stop the first lecture, and we'll pick up video 2 with the non-phagocytic killing mechanisms.